Okay, good afternoon. All right, the only announcement I have before we get started today is that I just want to encourage you to get going on that chapter 5 metabolism homework. If you haven't done that already, get that out of the way. The homework assignment with the chapter 7 genetics is a little bit longer because there are some animations to watch that I think that are really good. Take the time to get all that you can out of that. And then there's only one homework assignment before the next exam. We're going to be doing viruses next week. So I know the exams, they start coming fast and furious. It's not next week, but the week after our next exam. So, you know, gotta just kind of keep chipping away at it, right? Don't leave it all till the last minute. All right, so last time we talked about the structure of DNA and how it is replicated, kind of big picture, semi-conservatively. You should understand what that means. And we also talked about all the enzymes involved. Like, you're gonna want to recognize all those enzymes like DNA gyrase, DNA helicase, DNA polymerase 3, ligase. So understand what all those enzymes do as part of replication. We talked about RNA in general and some of the different kinds of RNA before we started on transcription. So that's how RNA is made from DNA. All right, so we ended at kind of a crucial time, the initiation of transcription. So I brought in just a couple other slides that are not in your book, just to talk through some details a little bit more here about initiation. So I already, you know, kind of wrote quite a bit on this slide, but just to review, here's the DNA strand, and there's a promoter region upstream of where the transcription is actually going to start. Okay, and that's where the RNA polymerase needs to bind. All right, and the more tightly the RNA polymerase binds to that promoter region, the more likely it's actually going to transcribe the gene. All right, if it only attaches kind of loosely, then it might fall off and not actually transcribe. Now, sometimes maybe that's what the cell wants. Maybe it's going to encode for a protein that isn't needed all that often. Okay, there are definitely some regulation that happens there that we'll talk about a little bit later. But this sigma factor, this is a protein that will bind to the RNA polymerase and help it, it will associate with the RNA polymerase to help it bind to that promoter. And the other regions that we talked about, this -10 base pair region and the -35 region. So when we say minus, that means upstream of where transcription starts. We think those are two regions that are especially important for the RNA polymerase to recognize to bind tightly. And remember, we had a special name for this -10 region, the Pribnow box. Okay, and that's, assume that I'm always talking about bacterial transcription. We will talk about some differences between eukaryotic and bacterial transcription, but assume that we're talking bacterial here. Okay, so these, I want to take a little closer look at these two regions, the -10 and the -35 region. So with this slide that I will admit looks enormously complicated, I just want to use this to point out a few things that will hopefully kind of help you see the bigger picture. So what all these letters are, these are nucleotide sequences of different promoter regions of operons. Okay, so remember, an operon is a group of genes under the control of the single promoter. And we're going to talk more about operons soon. But so this just lists, there's actually, we figured out between like 600 and 700 different operons in E. coli alone. So this is just a list of some of the sequences of those promoter regions in different operons. All right, so ignore most of them. The Lac Operon, that's the one that people are most familiar with. So if we just look at those sequence of nucleotides, again, this is in the DNA in the promoter region. And if you look at that -10 region, that's highlighted in the -35 region. You know, just remember, this is what the sequence of bases. And we know that after we've sequenced many different operons, we've noticed that there are common themes in those two regions. So if we looked at all those regions, we've noticed that there's some consensus sequence in the -35 and the -10 regions. So in other words, most often here, that first base is a T, 77% of the time. And then 76% of the time, the next one is an A. All right, so we understand that the closer the promoter region of the Lac Operon is to that consensus sequence, the more likely RNA polymerase is to bind. All right, so that's just what I'm trying to take home here. So the closer the promoter sequence is to some consensus sequence, again, the more likely RNA polymerase will bind. And again, the more likely it binds to the promoter, the more likely then it transcribes the gene into RNA. Okay. So basically, that's the initiation step. So the beginning of transcription. Okay, once that RNA polymerase binds and starts moving along the DNA, then we call that elongation. So this is a graphic from your book here. There's not a lot to say here. Here's the sigma factor. So sigma is falling off. Its job is done. Its job is just to get that RNA polymerase to bind to the promoter region. Once it does that, it falls off. And you can see the new transcript here is in red, and you can see it's adding on to the 3' end. Okay, and again, those ribonucleotides are carrying with them the energy to polymerize into the RNA. All right, so 3' hydroxyl group adding on there. And moves down the DNA. Remember that RNA polymerase melts or unzips those 2 strands of DNA itself. It doesn't need another enzyme to do that for it. All right, so that's elongation. Just keeps going. Pretty simple. And then just to mention that this can be happening like multiple RNA polymerases can be binding to that DNA and moving down it at the same time. So you don't have to wait till one new transcript is made and then start another one. Lots of RNA polymerase enzymes can be binding at the same time. Okay, and we think that happens in both bacteria and eukaryotes, both. Okay, and especially if it's, you know, some encoding for some protein that the cell needs a lot of. Okay. All right, so how does transcription stop? Remember, replication is easy. All the DNA is copied. There doesn't really need to be a set, you know, signals for it to stop. Transcription, only part of that DNA usually is going to be made into RNA. So we know there's very specific ways that transcription termination comes about. And there's two different methods that happens in bacteria, and we think that they happen with pretty equal frequency. Okay, so the first method is known as Rho Dependence Termination. So basically, this involves a protein called Rho. And Rho is actually a helicase enzyme. So remember what a helicase did during replication? It's the enzyme that melded the two strands apart or unzipped. So basically here, this little pink bracelet almost is the Rho protein. We think that moves along the new strand of RNA and basically will kind of unzip these kind of, the RNA only stays kind of transiently bound to DNA for a short period of time, but that RNA will move along and kind of release the RNA from the DNA and dissociate. This is the RNA polymerase from the DNA, and kind of all the parts fall off. Okay, so you know, this method, this leaves a lot of questions in my mind, like, well, how does the Rho protein know exactly when that this should happen? I've looked more deeply into this, and two things. Number one, it gets complicated really, really quickly. So it's kind of nice that this is just the way the book presents it. And number two, there's a lot that we don't understand about exactly how this happens. So we know that one method of termination involves the Rho protein. And that's basically all you need to know about that method. The second method does not involve Rho protein. So we have a couple different names for that. We can call it Rho independence, which does not involve Rho, or sometimes it's called sequence dependent or self-termination. Okay, that's all names for the same thing. Basically, with this method, there are palindromic sequences in the DNA. So a palindrome, it means it's, you know, going the opposite direction in the other way. So basically, we've got these, for example, it's usually rich in G's and C's, and the new transcript kind of lines up because it's the same one way. It's hard for me to explain this and back the other way. These will actually complementary bind together and form this hairpin loop. So again, like, anyway, if you want me to go deeper into the palindrome thing, I can sketch that out a little bit later, but I think it's not super important there. But it will form this hairpin loop that causes some mechanical stress that causes the transcript to fall off of the DNA. And in addition, there's usually a sequence that's rich in A's and U's here. So remember those A's and U's, they only have 2 hydrogen bonds rather than the G's and C's that have 3 hydrogen bonds. So this dissociates a little bit easier when you have a stretch that's rich in those A's and U's. Okay, so again, basically that hairpin loop creates tension, and that mechanical stress kind of makes the transcript fall off. All right, so again, those are the 2 ways that we think transcription ends. Probably about 50-50, you know, which way that happens. All right, so I do want to bring up a few differences in prokaryotic. It's more correct to say bacterial because there are some differences between bacteria and archaea and how they do their genetics. But anyway, prokaryotic and eukaryotic transcription. So thinking about where these processes take place, There is some differences here because in prokaryotes, this is going to be in the cytoplasm. Again, if you think about transcription is going to take place where the DNA is. All right, so it might be more correct to say the nucleoid region of the cytoplasm, but again, there's no membrane around that there. For eukaryotes, this is going to take place in the nucleus. Okay, that's where the DNA is, so that's where transcription is going to take place. Eukaryotic transcription is a lot more complicated. So there's a different RNA polymerase for each kind of RNA that's made. So there's one kind of RNA polymerase for transfer RNA and a different kind of RNA polymerase for ribosomal RNA. For bacteria, they just have one enzyme that makes any kind of RNA. And there's lots of these protein transcription factors that are necessary for initiation to begin and even for elongation to continue. So again, just lots more complexity for eukaryotes. And in addition, there's some processing that has to take place in eukaryotes after termination takes place and before that messenger RNA can get read at the ribosomes. Okay, so the first thing that happens again, and this is in eukaryotes, is that there is a modified guanine cap put on the 5' end of the messenger RNA. And we'll get to why that's important in just a minute. And then the tail is the 3' end is polyadenylated. Okay, so that's the 3' tail or the 3' end. Okay, so the 3' polyadenylation is basically a bunch of adenines added to the 3' end. Okay, that guanine cap is modified a little bit. It's actually, the guanines are methylated and have some unusual linkages there. But so basically, there's some modification that has to happen because the ends need to be protected. Because in eukaryotes, that messenger RNA has to travel from the nucleus out into the cytoplasm where the ribosomes are. All right, so there are nucleases in the cytoplasm that would chew up the ends of that RNA. So this is a mechanism to protect the ends because it has to travel a little bit. Okay. And the third thing that happens in eukaryotes is that there typically is some splicing that takes place. In other words, there's parts of that messenger RNA that are not necessary and get cut out. Okay, and I got a picture to show you that in a minute. But the exons, those, you could think of that as expressed genes. Okay, they are going to remain. And the introns, you could think about them as intervening sequences. Those get cut out. Okay, so exons are expressed, introns are gotten rid of. All right, so if you look at this book, this picture from your book is helpful in showing that, again, and this is eukaryotic messenger RNA. Here's what they call the pre-messenger RNA. So we've got the 5' cap has been added. Yes, question. Oh, that part right there. Yeah, okay, I'll make that a little smaller like that. Okay, so it's got the 5' cap. It's got the polyadenylated tail in the pre-RNA, and then it's got exons and introns so basically this spliceosome complex, a protein enzyme complex will come through and splice out those introns. So that's what's left and ready to leave the nucleus here is just the messenger RNA now that's just exons, but it's got the protection on the caps. Okay, so there is a video, an animation that's part of your homework that it's a great little tutorial and talks all the way through transcription and into translation, but it shows this. It talks about splicing and the polynylation, and it's not clear that that's eukaryotes. All right, so normally we're only interested in the bacteria, but just kind of be aware that it shows it leaving the nucleus and like, wait a minute, this is not a bacteria. So it's not terribly clear about that. All right, and just I pulled in another picture from another source here. Just be clear that can be kind of a level of regulation because cells can splice the same transcript a little bit different to get unique products, unique proteins here. So if that gene is sliced sort of how we would expect it to be, if those lines are introns here, it would look like this. Maybe though this whole gene exon here gets spliced out, so you're going to have a protein that looks a little bit different, or maybe part of an exon is spliced out, so you could get 3 different proteins from that same like pre-messenger RNA. Sometimes the cell does that on purpose. Sometimes errors are made in splicing. And we know that some diseases are caused by errors in splicing. Duchenne's muscular dystrophy, we think, is caused by errors in splicing. Perhaps Parkinson's, and there are others. So I'd bring that up. All right, so before we move forward and think about how that messenger RNA transcript can end up being translated into a protein, we need to have a little bit of understanding of the genetic code. All right, so this is not anything that you're going to memorize. What this is let me change the color here. Here we have the codons in messenger RNA, and how we know what bases correspond to what amino acids. Okay, so one messenger RNA codon and what amino acid it corresponds to. It took us a while to understand this code. Marshall Nirenberg was working at NIH there in Bethesda, Maryland in the late 50s, early 60s. And this was painstaking work to figure this out. It involved radioactive labeling and a lot of trial and error. We didn't even understand to begin with. We knew there was 4 bases, right? And then how did that correspond into the 21 amino acids? All right, if it was just a pair that corresponded, then there's only 16 possibilities of 4 nucleotide bases. But if you had triplets, then there was like 64 possible combination of those 4 amino acids. So we figured out that there is some redundancy in this code. In other words, sometimes it doesn't matter at all what that 3rd base is. Okay, so there is, again, some redundancy. Sometimes we refer to that as wobble in that 3rd base of the codon. Okay, oftentimes it doesn't really matter what that 3rd base is. It's still going to encode for the same amino acid. Okay, sometimes it does matter. Again, like here, if it's U or C, it's phenylalanine. If it's A or G, it's leucine, but sometimes it doesn't. Okay. Let's see, what else I want to say about this. So 21 amino acids, plus you have start codon, So that's the amino acid that always is going to begin or initiate translation. When the stop codons are read at the ribosome, then that's going to terminate translation. All right. So again, don't memorize all those. You know, probably the start codon should be familiar, but other than that, it's pretty universal throughout life, basically, with very few exceptions. All right, so when we think through the steps of translation, the first thing that we're not going to really spend any time on here, but in the past I've gone through this a little bit more carefully. The tRNAs are what bring the amino acids to the ribosomes to kind of match up with those instructions in the messenger RNA. Okay, so the process of the tRNA adding that amino acid, we call that charging. Okay, so a charged tRNA is one that carries the appropriate amino acid. Okay, so again, we call that tRNA charged. And there's really just some enzymes that facilitate that. It requires some energy to add the amino acid to the tRNA. And then this should look familiar, kind of the same way that we describe the steps of transcription: initiation, elongation, and termination. So you're just going to want to be careful as you're studying to keep transcription events kind of separate in your mind from translation. Again, transcription kind of sounds like translation, but keep those processes separate in your mind. They're very different. And then we'll just briefly mention that once you have a protein translated at the ribosome, that doesn't necessarily mean that it's ready to do its work. Okay, there might need to be some tweaks made before that's a functional protein. So we'll just mention that briefly. All right, so let's start with the graphics that they give you in your book about how translation is initiated. Okay, let's see. All right, so remember, this is the way your book writes the tRNA, kind of a little hooked structure there. And depending on, especially on the sequence of the anticodon of the tRNA, that's going to determine which amino acid it's carrying. Okay, so here's the messenger RNA. It has found a small subunit of the ribosome, and here it shows it already docked to that small subunit of the ribosome. We're going to look at that a little bit more closely in a minute, but that start codon is going to dock at the P site. So P typically stands for peptide or peptidal. That is where the growing polypeptide is kept or is held. Okay. The A site, we're going to, typically that's short for acceptor site, and that's where the new amino acids dock, except for the first amino acid. Okay, you can see that first amino acid actually docks right there at the P site. All the other ones are going to come in and land at that A site of the messenger RNA. Okay, and then there's another site, an E site, that stands for the exit site that we'll get to shortly. Okay, so the start codon, again, is lined up with that P site, the tRNA carrying the formal methionine, which is always the first amino acid. Any protein comes and matches and docks at that P site. Okay, the energy currency here that the cell uses is GTP instead of ATP. And then, so once that tRNA docks, then this forms what we call an initiation complex. So the large subunit of the ribosome now comes together. And remember, in bacteria, that's 50S, and the small subunit is 30S. Okay. So now we've got the initiation complex, we've got the first amino acid, and we're ready to go, ready for that protein to elongate. Okay, before we talk about elongation, I just want to point out a few things about initiation. Yeah, this happened to me before. My whole picture here didn't get exactly, I must not have copied that right. Okay. So I took this picture from a different source because I wanted to point out a few more details about the initiation here. First of all, orient yourself. This up here, it's actually showing the small subunit. Okay, so oftentimes in pictures that's drawn on the bottom and the large one on the top. For whatever reason, they drew the small subunit on top. And what's docking here, it's really important that the messenger RNA here lines up at just the right spot on that ribosome. So there's a special site on the ribosome that it docks, and that's actually part of that 16S strand of ribosomal RNA. That's part of the 30S small subunit. Again, there's a specific site there. We call that the messenger RNA binding site. And there's a specific few nucleotides on the messenger RNA that has to line up precisely with that site. And so we have a special name for that, the Shine-Dalgarno element. Now your book mentions that briefly, but doesn't really incorporate it into any of its graphics. So I wanted to bring that in. It's important that that lines up just right because this AUG has to line up just right with the P site. Okay, think about it. If this doesn't here line up just right, what if this is off by, you know, one base pair? All right, UGC, okay, that's going to encode for something totally different, and it's not a start codon. Okay, so initiation can't start unless that lines up just right. Try to erase that there. Okay. Okay. In eukaryotes, they don't have a Shine-Dalgarno element. They just line up, their 5' cap lines up just right here. So there is some differences there. How do you remember that this is the 5' end? This was part of my, this didn't really show up. I don't know if my drawing showed up on what I posted, but I was trying to show that RNA, remember this is messenger RNA, can still be, I don't even know if I can draw this again. Okay, here's the DNA. Okay, this RNA can still be transcribed. So if this is RNA polymerase, this could really be happening at the same time. Transcription isn't even done yet. And all of a sudden the ribosome is clamping onto one end of the transcript and starting translation. Okay, because this is all happening right in the cytoplasm for bacteria. Okay, so again, this transcript could still be getting made. Okay, and you know this is the 3' end of the transcript because it's always adding on to the 3' end. Okay, and so this is the 5' end. Translation can be starting even before transcription is finished. Okay, all right. So stop me if there's questions, but then in terms of just the growing peptide chain, the new ones just come in and enter at the A site, the tRNAs, the charged tRNAs with, you know, again, don't memorize what matches up with UUU, but here it happens to be phenylalanine. Those that enter into the A sites. There are peptide bonds then that are formed by ribozyme. Remember, ribozyme, this is ribosomal RNA that acts as an enzyme. within that ribosome there somewhere. Okay, so forms a peptide bond there between those two amino acids, and then it shifts into the P site. So whether you think about the messenger RNA kind of pulling through or the ribosome shifting, either way, and then so that growing peptide is considered to be kind of sitting in that P site. Okay, add another one. Here comes threonine, adding into the A site. Again, ribosomes catalyze peptide bond formation there. And then that the next tRNA, now that's uncharged, leaves from the exosite. And I found a good YouTube video here. I'm trying to remember. If you start that, it goes through transcription a little bit first, and then at about 2 minutes, it will start showing, you know, a drawing basically of what animation looks like, or sorry, of what translation looks like. And it's really quite interesting. You really kind of learn to appreciate the complexity of this and how fast it happens. How fast does it happen? About 15 amino acids are added per second. All right, so it's happening enormously quickly. Hard to understand how things can happen that fast, but again, that video kind of helps you appreciate the speed of that. Okay, and understand that we can have lots of ribosomes attached to that messenger RNA at any one time. Okay, so as soon as that 5' end pulls through, you know, another ribosome can attach there, and another one, and another one, and another one. Now, messenger RNA actually gets degraded pretty quickly. So eventually, you know, it's not going to just keep making protein forever and ever. If it needs more protein, the cell needs to just keep making more messenger RNA. Okay. And we think this happens a little bit more often in prokaryotes than what it does in eukaryotes for whatever reason. All right, so how does translation end? Your book, I didn't really like the little graphic that your book had. There was a mistake, kind of a minor error in it. I can't remember exactly what that was now. So I pulled in a graphic from a different site. So basically, once a stop codon comes to that A site, so UAA is actually a stop codon. There's not any amino acid or tRNA that's going to recognize that. And so that pause causes these protein release factors to come in and bind to that A site. Okay, and that's going to activate the ribozymes in the large subunit to sever that growing polypeptide from the tRNA. Okay, so you can see here how that polypeptide chain has been released when those release factors come in. And thirdly, then the ribosome dissociates. Okay, the small subunit comes apart from the large subunit. Okay, release factors are released. Okay, and translation stops. Okay, so for transcription, termination either involves a Rho protein or it's dependent on some sequence that's read. Translation stopping involves a stop codon and those release factors. Okay, so very different processes. Now, is that protein ready to go? Not necessarily. It depends what the protein is, of course, that's being made. Very often in bacteria, the first thing that happens is that formal methionine is cleaved off. And I'm not sure why that happens, other than if it's a bacteria that may likely be infecting an animal or a human. That's a big tip off to our immune system. If formylmethionine is red, hey, that's something that is not us, right? So our immune system is going to recognize that, and again, it might be not help the survival of the bacteria if our immune system notices that formylmethionine. If it's some kind of enzyme, then it might need some kind of cofactor in order to be functional. So those or cofactor or coenzyme might need to be attached. There might need to be some complex bonds made so that the protein can fold correctly. And for bacteria, it may be something like a toxin. So that bacteria does not want that toxin to be functional as soon as it's made. It might be some kind of trimming that happens right before it's released from the bacteria cell, so that, again, it's something that acts outside the bacteria and not within the cell. Okay, so it's in the best interest usually, or it can be, for it to not be functional right away. All right, so I like this picture from your book, and they kind of simplified this for the latest edition, and I'm not really sure why, but I think it kind of it helps maybe kind of bring it all together. All right, so that template DNA strand, remember, in bacteria that DNA can be polycistronic. So in one basically transcript, you can have information for several different genes, polycistronic, many genes that are transcribed together. Okay, that's essentially what an operon is, right? We'll get to that. And so that messenger RNA will actually have encoded into it a shine Delgarno-site. That's going to line up with the ribosome. It's going to have a start codon, a stop codon, and that's going to eventually be one protein. Okay, and then it will have all of that for its second protein and its third protein. So kind of helps you visualize the whole process a little bit better, I think. Okay, so just to sum up, I think I probably mentioned all these things already, but just to sum up, and really it's better here to say bacteria, because that's not necessarily true for some archaea, some of these things. But we know that in eukaryotes, there's always going to be one protein come from one gene, where bacteria, like I just mentioned, can be polycistronic. In eukaryotes, we talked about that 5' cap that's necessary to protect that messenger RNA as it travels. That also serves to line up at the ribosome to initiate translation, where bacteria have what's called a shine Delgarno sequence, in order to, we'll say, match up with the small subunits of the ribosome. For eukaryotes, the first amino acid is always methionine, and bacteria have a formal group added to that, so they always have formal methionine. I tried to look up, why that is, and again, other than the I don't really know why that would be the case. And like I mentioned, transcription is going to be in the nucleus of eukaryotes and then travels to the ribosomes and the cytoplasm. Transcription and translation are both taking place in the cytoplasm for bacteria. Okay. All right, and I just wanted to draw your attention to a table in the book. Again, sometimes these tables are useful to kind of quiz yourself. So As you look through this, cover up part of it, and then, see if you know. This is just basically the basics of replication, transcription, and translation. But that's just a common thing for students to get all three of those processes mixed up. Yes. Oh, yep, didn't finish writing in the last one. Again, I could finish my thought here. Cytoplasm, transcription, and translation takes place in the cytoplasm. That's what I meant to say there. I just didn't write it all out. Yes. Oh, yes. So sorry. This stands for formal methionine. Okay, that's just the first amino. That's always the first amino acid in any bacterial polypeptide. All right. So again, this is good to quiz yourself. I would say we didn't really talk too much about fidelity mechanisms. So that means what mechanisms are there to make sure it's done correctly. We did talk about that a little bit with replication and how that DNA polymerase 3 has some proofreading to make sure it's putting in the right bases. The fidelity is highest for replication. There are more mistakes made in transcription and even more mistakes made in translation, but that's not as crucial, because as long as the blueprint is right, the next time hopefully those proteins will be made correctly. Question? Yes. So when I say fidelity, that means how likely it is that there were no errors made. So replication, we want that to have the highest fidelity. Okay, if we make a mistake in the blueprints, forever after, every protein that's made is going to be wrong. So that's a huge problem. We have we're going to talk about ways that cells can fix errors, in fact, in their DNA. But if there are errors in transcription or translation, that's not as big a deal, because probably the next time it's made, it will be made correctly. So we see even more errors in translation than what we do in transcription. Sometimes the tRNAs are charged with the incorrect amino acid. Again, that's not a huge problem. The other thing, we didn't really talk about this, so I wouldn't worry about kind of the direction that the amino acids kind of are bonded with each other. We didn't really get into that. Everything else there, again, is good to kind of quiz yourself and make sure that you got the basics straight. Okay, so we're gonna switch gears a little bit, but not too much. Don't forget what we just talked about. Thinking about regulation. So why would a cell care like how much of a protein it makes of something? It's to conserve energy. Okay, a cell obviously doesn't want to be making a bunch of proteins that it doesn't really need. Okay, do you know how many ATP it costs a cell to make an average? Okay, so an average protein of about 300 amino acids. Again, that's the average protein. Okay, I want you to turn to your neighbor and just guess how many ATPs it costs a cell to make an average protein. Okay, just guess to somebody, and then I'll tell you. Okay. Okay. Anybody want to throw out a guess? Anybody know? Okay. It's about 1,200 ATP. Okay. So, all right. So, brag to your neighbor if you were closer than they were. All right. To me, that seems really expensive. When you talk about, from one glucose, a bacterial cell may only get 2 ATP if they're doing some fermentation pathway. So a cell has to really take in an enormous amounts of catabolites in order to produce that much ATP. Okay, so obviously, if it's not absolutely crucial that it makes a certain protein, then it's not gonna bother, and it wants to have a way to stop making proteins that it doesn't need. Okay, so there's different kinds of regulation. We talked about this briefly at the beginning of class, consensus sequence variation of promoter regions. So that goes back, I didn't post this again, that goes back to this slide. All right, so depending on these regions right here, if the cell has it's a little bit different from the consensus sequence, or if it's really close to the consensus sequence, that transcript is almost all the time going to be transcribed. But if it's a little bit further away from it, less often. If it's very different, then not very commonly transcribed at all. So depending on what the protein is, you know, the cell can kind of vary that somewhat. We know that there are different sigma factors that the RNA polymerase can use that is more likely to make it bind, or maybe less likely to make it bind. So again, that's another way that, you know, sometimes DNA is transcribed and sometimes it's not. But now we're going to spend a little time talking about operons, because again, these are genes that are regulated together. And again, this is a bacterial thing. We know that eukaryotes have some gene clustering, but for them to regulate a bunch of genes together appears to be a really rare thing. Question? So it is RNA polymerase. Okay, so more or less likely for RNA polymerase to bind. Okay, so that's in the initiation of transcription, but that's important there. Okay, so operons are really an ingenious way for a cell. If it's got, for instance, maybe 4 different proteins or enzymes that are necessary for one pathway to happen, it just makes sense that you're going to want kind of the same amount of those proteins. So regulate the amount of those together. It's a very efficient way. Okay, so thinking about what's really going on with operons, this is just basically, don't forget everything that we just talked about with the transcription initiation, because that's basically what we're talking about here. We draw out a template DNA strand. There sometimes is a regulatory gene, maybe upstream here, that would encode for some kind of repressor protein. The promoter region, remember, this is where the RNA polymerase binds. Okay. And if there is some kind of repressor, it is going to bind to the operator region. Okay, so don't necessarily memorize that. I'm going to show you why that makes sense in a minute. And then in order to really understand what's going on with a particular operon, you have to understand why the cell would want to make these genes. Okay, so I'm going to give you a few examples of that. If you kind of think like a bacteria, I know that sounds kind of corny, then you will help you understand and sort of reason through what's going on here. All right, so there are a couple of ways that we like to categorize operons. I'm going to give you a couple of examples of operons that are going to fall under negative control. And then one that falls under positive control. So your book kind of mentions those things, but it doesn't kind of categorize things quite the same way. So if an operon is under negative control, that just means that there is some kind of repressor protein that's important to determine whether the gene gets transcribed or not. All right. And like I just mentioned, the repressors always bind to the operator region. Again, I'm going to show why that makes sense in just a minute. So 2 types of negative control. I'm going to give you an example of each one. Some operons fall under what we call induction. Okay. And that just means this just means that the operon is typically off. Okay, so what I mean by that, the operon is not typically transcribed. And that's what I mean by being off. Okay, or maybe it'd be easier to understand if I say the DNA is not typically transcribed in an inducible operon. And usually a catabolic pathway is going to be regulated that way. So that means when you have some enzymes that are necessary to break down some sugar, for example, that's typically the way that they're regulated is through an inducible operon. Okay, so there's going to be some inducer present that deactivates a repressor. Okay, and the example that we're going to show here is the Lac Operon. So I'm going to look at that in just a minute. A repressible operon, then this is usually transcribed. So in other words, it's usually going to be on. And then only if there's some kind of co-repressor does it get turned off. All right, and usually anabolic pathways are going to be regulated this way. So pathways where the enzymes are going to catalyze the production of some molecule. All right, and the example that we're going to look at here for this is the trip operon. Okay. All right, so hopefully, I mean, I'm sure you've talked about Lac Operon at least some point in your career here. I want to kind of go through this rather quickly. Oftentimes in the past, I've stopped and had students see if they could label all the parts here. Well, I realized when I went through your homework, you do the exact same thing in your homework. All right, so I'm going to go through this a little bit quickly. Stop me if you know, whatever, you have questions. But we can go through and label the parts of this. So, you know, keep in mind what the cell is trying to accomplish. All right, these 3 structural genes are encoding for enzymes that are necessary to do what? To metabolize lactose. Okay, one of the enzymes breaks that disaccharide into its 2 monosaccharides. One of those proteins is necessary to help the lactose get into the cell really quickly. It's like a fast pass. If you've driven in Chicago and you have a fast pass, you go right through the toll booth. You don't even need to slow down. Okay, if you don't have a fast pass, you have to stop and pay the toll, and you pay a little bit more. Okay, so that one enzyme, a permease enzyme, lets lactose get into the cell really quickly. It can get in other ways, kind of slowly, but it can get in a lot quicker that way. This third protein might have some kind of detoxification effect. We don't really understand that as well. But those proteins, again, are necessary for the organism, the cell, to use lactose. All right, so it's only going to want to make those if lactose is present. Okay, but in the top here, there's no lactose. Okay, so here's number one. This is the RNA polymerase. It's bound here to the promoter. But there is a repressor here. And that's bound to the operator region. Okay, can the RNA polymerase get by here in order to transcribe the DNA? No, because there's a repressor in the way blocking it. Okay, so the repressor, it doesn't make sense for it to bind at the promoter region. It always binds at that operator region, which is downstream there. Okay, so no lactose. You're not getting these enzymes, and so that makes sense because there's no lactose there. So in the bottom here, these little floopy pluses, okay, now there's lots of lactose in the cell. Okay, so it binds to the repressor, okay, to remove that from the operator region, and now the RNA polymerase can go through and transcribe those proteins here that are necessary to catabolize lactose. Okay, makes sense. It's actually not quite that straightforward because there's another layer there. Well, what if glucose is present as well? The cell is going to always choose to use glucose first because that's a lot easier for it to metabolize. So we'll get to that in a minute. There's another layer we have to add to that. But first, let's look at another example of negative control, and that's the trip operon. Okay, so this trip stands for tryptophan. So the point of these enzymes here that are made, they are necessary for the cell to manufacture the amino acid tryptophan. Okay, so that's an amino acid that the cell usually needs. So that pathway is usually going to be on. So again, once it makes those enzymes, then the cell can manufacture its own tryptophan. But what if we put those cells in some medium where we've already provided tryptophan for them? Okay, it doesn't make sense for them to just keep making their own. Okay, that would be wasting energy. So we found that tryptophan actually will bind to a repressor that before was inactive, but now that it's bound with tryptophan, that activates the repressor. Okay, and now it binds to the operator region here and transcription gets blocked. Okay, so if there's lots of tryptophan around, we'll activate the repressor and now it doesn't need to make any more, so it stops. Okay, so again, we consider it negative control because it involves a repressor. Whether the repressor is usually there or whether the repressor is usually off, both cases involve a repressor. Okay, let's come back to the Lac Operon and talk about that extra layer, that positive control. Oh, and by the way, I think that I don't see, maybe I just didn't post it on mine, but I did hopefully post a YouTube video that talks about the trip operon. Okay, if I didn't put it on there, then somebody emailed me about it, and maybe I just didn't get that pasted in there, right? So anyway, it's always nice to see things, talked through again, maybe a little different way. Okay, so then the other layer of the lactose operon. So like I mentioned, if there's glucose in the media and lactose, the cell doesn't want to be making the enzymes to catabolize lactose because it would rather just use glucose. It's just more apparently easier to use. So the cell's always going to use that first. So there's this other layer of positive control on here where there's some sort of activator that has to be present in order for the DNA to get transcribed. Okay, so in this case, we're looking at the levels of cyclic AMP in the media. Okay, but let me first write glucose must be absent for cell to utilize lactose. Okay, so let's just make sure we got that straight. Okay, so the levels of cyclic AMP in the medium are going to be inversely proportional to the levels of glucose. Okay, and when there's lots of cyclic AMP, it's going to bind to this CAP protein. That stands for catabolite activator protein. Glucose and lactose are catabolites. There's something that's catabolized. Okay, so when there's lots of cyclic AMP, it will bind to that CAP protein here, and the RNA polymerase recognizes that, and only when it recognizes that complex will it transcribe the lactose operon. Okay, so if glucose levels are high, then cyclic AMP is low. All right, so you don't have much of that cyclic AMP. It's not going to bind to the CAP protein, and you're not going to get transcription. Okay, so no transcription of the Lac Operon. Okay, so if there's lots of glucose, the cell doesn't want to bother to use lactose. Okay, however, if glucose levels are low, then cyclic AMP levels are high. All right, and then you get lots of this cyclic AMP CAP protein complex that activates basically the Lac Operon. So now those enzymes that are necessary to metabolize lactose are going to be transcribed. Okay, and the picture from your book, I think, does a really nice job, once you understand that, of visualizing that a little bit better. So here's the CAMP, cyclic AMP bound to the CAP protein. The RNA polymerase actually recognizes that. So it's kind of hard to understand why something upstream would make a difference, whether the RNA polymerase binds or not. But this is a big enzyme. It recognizes that. And only when it recognizes it, now it will transcribe those LAC genes, okay, that are necessary. Yes, question.
So in basic terms, negative control involves the repressive protein and positive control involves an activator.
Exactly. Okay, so negative control involves a repressor, positive control involves some kind of activator. Yeah.
So does the LAC gene, does that transcribe the enzyme to break down lactose?
Yes.
So
yes.
If there is a lot of glucose, the LAC gene won't get transcribed, it won't be active.
Exactly.
But that doesn't happen via a repressive protein when there's glucose.
Right. So there's still the level, I mean, there's still the Lac Operon is still kind of in go, like if there's no lactose and there's no glucose, right, it's still not going to be transcribed. If there's lactose and no glucose, then it will get transcribed. So I think you might have said it correctly, but I would have to listen to you a little bit more carefully. Okay, so again, these LAC genes are necessary to break that disaccharide into its monosaccharides and help it get into the cell more efficiently. Okay, This is an excellent YouTube video. It's about 5 minutes long that talks about both the negative control of the Lac Operon and the positive control. Okay, so again, the real key is think about why a cell, when a cell would want to be able to break down lactose and when it doesn't need to be. Okay, another question.
So for the RNA polymerase to transcribe, it needs the lactose to bind to the repressor and the loop in the operator plus this to bind to that binding site.
Right, it needs both things, right? Lactose has to bind to the repressor as well as the cyclic AMP and the cap protein. Okay, good. All right, so what evidence do we have that the Lac Operon is under both of those methods of control? Well, if we inoculate a test tube with some bacteria and the media has both glucose and lactose in it. Sorry, my handwriting's bad there. Okay, so we inoculate an organism into a broth test tube that has both glucose and lactose, and we count the cells in that test tube by means of the easiest way is with a spectrophotometer. All right, so we look at the cloudiness of it over time in order to construct a growth curve. Okay, so normally a normal growth curve has kind of a lag period and then a log period where it's growing exponentially and it evens out into a stationary phase, right? That's normally what we see. But if we, again, have a medium with glucose and lactose, we actually see evidence that one catabolite is being kept from being used, all right? So in other words, lactose is not used at first, that's repressed, and then eventually is used. So we actually see a lag period to begin with, and a log period there when glucose is utilized. Once glucose is run out, then we see another lag, but then we get another exponential growth period here when lactose is utilized. Okay, so we refer to this as dioxic growth, and that just means two-phased growth. Okay, we actually get two different log periods and two lag periods. Okay, so that's how we know that the organism is using one metabolite and then it's switching to another. And in the process of switching over, you get a little bit of a lag where it's ramping up the enzymes in order to use lactose. Okay, so that's kind of cool to do in the lab. It takes a while to actually see that if you're using E. coli, but it's kind of cool to do. This seems like maybe a good place to stop. So we have just a little bit more of the genetics that we're going to finish next week. We're going to talk about mutation a little bit and horizontal gene transfer, and then we are on to viruses. All right. So have a good weekend.